Yeah, it's a pleasure to be here, and thank you for invitation of Standard BioTools. So today I would like to present our work in brain tumor biology and how also we implemented presented methods to our research. So as mentioned, I'm coming from Ljubljana, Slovenia. Today I have no disclosures. So today I would like to present you the work we are doing for the past few years. The advances of cancer treatment are enormous in recent years. There have been huge advancement in tumor diagnosis, also early screening and also treatment, but still we have a lot of challenges. So we have poor outcome of patients with aggressive tumors. One example is brain tumor that I will talk about it today. And we have pancreatic and ovarian that are very aggressive. And there's still no efficient treatment for those tumors. Then a lot of tumors resist to the therapies. We have different chemotherapy approaches and also targeted approaches that can be efficient for some time, but then the tumor usually regrows or reappears. And most of the new drugs, it's estimated that around 90% of the new drugs fail clinical trials. So we still have a lot of work to improve all those segments. And why this is happening, like those reasons that I just mentioned. One is that the cancer is diverse disease, so it's highly heterogeneous across the patients and within the patients, although they may have the same diagnosis, they can be very different in terms of cell composition, in terms of molecular heterogeneity. They also adapt to the treatment, so we call them that they are plastic. Then we actually have unexplored biomarkers that can predict whether the patient will live longer or will have poor bad prognosis or will respond to treatment. And the tumor models in the laboratory do not reflect the conditions in patients. So we have tumor models that not reflect the tumor complexity in terms of the microenvironment and also the molecular heterogeneity. So there are still a lot of challenges here. And for us, it's very crucial that we want to target and target more efficiently glioblastoma. So it's the most deadliest, it's one of the deadliest malignancies. It's a brain tumor that arises from astrocytes, and it's highly aggressive. We have around 1.5 years average prognosis after the diagnosis of these patients. And still after like 20 years of treatment with chemo, radiation, and surgical resection of the tumors, there are no significant new treatments available for these patients, at least that they would show some significant clinical benefit for those patients. And what are the reasons? The reasons are heterogeneity, across the patients and within the same patient. So we have this tumor microenvironment, which contains different tumor cells, but also other cells like immune cells, supportive cells, endothelial cells, stromal cells. So it's highly heterogeneous. Then we have these invasive cancer cells that can invade from the original tumor bulk up to a few centimeters out of the tumor, and it's really hard to remove them from the patient's brain. So it means that they stay in the brain tissue after the surgery and can cause the regrowth of the tumor. And those cancer cells, the glioblastoma cancer cells, are highly resistant to therapies. So it means that after the radiation and chemotherapy, those cells survive in the brain tissue, and after some time, few months, they regrow the tumor, which is most of the time very aggressive, and this is the cause of the patient's death. So there is urgent need to understand the molecular basis of the glioblastoma and also explore some new therapeutic approaches, including biomarkers. And today, I would like to show you the results of two publications that were published last year and this year. The aim of this study was to identify biomarkers for improved prediction of glioblastoma survival, then to explore distinct molecular signatures across tumor regions to identify these invasive cancer cell signatures, and establish personalized tumor models for translational studies and drug screening. So the basis for our research is our Gliobank that is based in Ljubljana in Slovenia and contains the biological material and data of Slovenian patient population. So we are collecting the brain tumor tissue, also the blood and the data, together with the clinical partners, so Institute of Pathology at Faculty of Medicine at University of Ljubljana, then Department of Neurosurgery from University Medical Center in Ljubljana, and Institute of Oncology in Ljubljana. And at NIB, at our institution, we are collecting the biological material, we are processing it, and we are obtaining different cell cultures. We are freezing down the patient tissue to enable us the translational research. So, for example, this is just an example of what we have, like from the cell culture point of view. We have primary glioblastoma cell lines. We have the stem-like cell lines, and also glioblastoma organoids that then we can treat in the culture and explore. And I will also show you this part in next slides. And the main methodology that we used for our research in this part of the study was Biomark system. So this is gene expression real-time PCR that is available at our institution. We were interested in this system because it requires low amount of RNA of our samples, because we are working with tissues, with the cells that are very precious. This approach also enables a targeted approach with high number of samples and also targets in a single experimental run. Based of what we have established at NIP, we thought it's a really straightforward protocol and the data analysis, so you can really quickly obtain the results. So this was these were the main reasons why we choose this platform to study the gene expression in our samples, and then link this data with the clinical data. And we use this integrated fluid circuit dynamic arrays that enables us to analyze several targets and several samples. So what was the workflow? So we started with we are starting with a tumor biopsy that we receive from the patients out of the clinic, so immediately after the surgery. Then we process those tissue pieces, we snap frozen some of the tissues, we isolate the cells or organoids. We can then also treat those cell cultures and then analyze different endpoints. So here we were interested in gene expression analysis. We did RNA isolation out of the samples. We then did reverse transcription, preamplification, and then we run biomark as mentioned in previous slides. Analysis was then done with the software and QuantGenius program or software that was also established at our institution at the FITO department. So we use the system to analyze our data. And everything is also written in our publication if you're interested for more. So the markers that we use were very targeted. So it was very targeted approach. We were interested in specific targets. like targets, like markers of glioblastoma cells, glioblastoma stem-like cells, the glioblastoma subtypes, so the markers that they are specific for subtypes of glioblastoma. Then we were interested in markers of mesenchymal subtype and epithelial to mesenchymal transition. We use some markers of immune cells, immunosuppression, and cytokines, and DNA damage response, because we also treat those samples and then analyze the response to treatment. And I will show you some results that we obtained. We actually identified the new independent prognostic marker that predicts the survival of glioblastoma patients. So high expression of DAP2, this is one gene that is increased in our samples. So if this gene was upregulated or highly expressed in our tissues, the survival was significantly shorter for glioblastoma patients, and this was independent of therapy, of age, of performance status. And we were one of the first that identified this marker associated with the survival. And this marker we also checked in our tissues and cancer cells and non-brain tumor tissue, and we could really see high expression in glioma of higher grades compared to lower grade gliomas, higher expression in glioblastoma cells, and also low expression in non-tumor brain tissue. And here we used around 100, just to give you approximate numbers, around 100 de novo glioblastoma patients. We were also, as I mentioned, we were also interested in these glioblastoma subtypes. We were interested in subtyping glioblastoma patients based on molecular signature. So we determined the markers and then subgroup those glioblastoma patients, and we could see that we could really make a good subgrouping, so really distinguish between different subgroups like mesenchymal, classical, and preneural, and the mixed type, so a combination of all three. And what we saw is that those patients that were subtyped really have different prognosis. So here is example that the mixed subtype population has the highest survival rate among the patients. And here we used like the machine learning method to distinguish between subtypes of glioblastoma. So we have like a method based on machine learning that can predict the survival of patients. And we were also interested in those invasive cells, so those cells that invade far away from the primary tumor mass, as you see here on the slide. And those cells usually survive the treatment. They're invasive, and we are interested what are their molecular characteristics. So if we know their biology, the molecular cell biology, it's a high chance that we can then also target them or develop some targeted approaches against those cells. And here we have a great collaboration with the University Medical Center and Department of Neurosurgery. So they could sample the tumors based on different regions, so like the tumor core and tumor edge. And then we receive those regions separately in our lab, so we can analyze them and see what are the differences. And we actually found a lot of like molecular differences in those regions, especially in subtypes. So we have proneural subtypes genes that are enriched in these invasive tumor areas. So in those areas that usually are responsible for tumor recurrence. And you can see here on the slide some examples of genes like HER3 or STM4 that are upregulated in this tumor invasive area or tumor rim samples. And usually the mesenchymal genes and EMT transition genes were downregulated or are downregulated in our invasive tumor areas. And this is also somehow in line with the literature that we read. So it shows the similar trend that also some research group in the past has already also found that. So this was the first part that is connected directly to the tumor tissues. But then we explored the molecular signatures and targeted gene expression also in our models, the organoid models. So those organoids are preclinical models that can be a critical step in cancer research and also drug screening. As I mentioned, most of the drugs that are tested in clinical studies fail. And this is partly due to the inappropriate tumor models that do not really reflect the tumor biology of the patients. And we have different kind of models like in vitro, ex vivo, and in vivo models. And those models are not perfect, but we are trying to mimic the tumors in patients as close as possible to their nature. So we use organoids. They can be used like a platform to test novel or combinational approaches for early evaluation of therapeutics. So here we have a models in the lab, and we can play with the condition. We can try different combinations like chemo, radiation, targeted approaches, immunotherapy, and that's why this platform is really nice, and also has a great potential to really early evaluate the potential of some drugs to be tested then on humans or mouse models. They are timely and high throughput methods, and they can contribute to a decreased number of experiments in animals. And we know that immunotherapy is really specific in a way that requires species-specific tumor immune cell interactions. So we can also play with immunotherapy because we have also immune component in our organoid system, and I will show you that later. So our platform is designed to receive the tumor tissue of the patients, then isolate and prepare the organoids that are treated in the lab, analyze with different methodologies. Like today, I will show you the PCR, and then those data can be linked with the patient data and their response to treatment. So what we did is we did the organoids from tumor tissue, so from fresh tumor tissue samples that we are obtaining in the lab immediately after surgery. So the tumor tissue pieces are cut in small pieces. They are then put in the right media, and then they are grown for several weeks. So here we can see example for up to 5 weeks, and those organoids are growing in the culture. So we are very happy how they behave and how they are growing. And then we checked like different proteins and genes in those organoids, if they mimic the original patient tumors, if they contain different tumor cell types like tumor cells, immune cells. So firstly, we did like simple immunofluorescence to check like the composition of the cells. We saw that they contain the tumor cells, but they also contain the endothelial cell markers, so the markers for the vessels, the immune cells like the macrophages like CD68 and EBA1. So those are all the markers of the macrophages and microglia. And besides that, we could also observe in a lot of organoids also the T cells, so the CD3 population, and we could detect the interaction of immune checkpoint inhibitors, and so the ligands and receptors like PD-L1 and PD-1. And then we went further, so to analyze the gene expression levels, because with that we could analyze more genes, and targeted. We could do targeted gene expression, and here we could actually saw that the gene expression profile of the organoids reflects the gene expression profile of parental tissue. So we could detect those cancer cell markers, cancer stem-like genes, also immune cells, subtype genes, DNA damage response genes. So we could detect also the same clusters of genes that mark specific cell population. And when we treated those organoids with radiation and chemotherapy, so this is the radio and chemotherapy are the standard treatments for those patients, we could see like mixed responses. We checked cell survival of cells, and also we checked the invasiveness, so how fast they spread out of the organoids. And we could see like mixed responses, but mostly like overall, when we did like the statistics, we couldn't see like really the effect of the therapy. So it looks like those cells in organoids are very resistant to radiation and temozolomide. But yes, some organoids from some patients did response, but overall there was no effect, and this is somehow correlating with the patient response in the clinics. And we were interested what are the mechanisms of therapy resistance. And we went again, we checked everything with targeted gene expression with Biomark. We choose different genes, and what we notice is increased expression of DNA damage response genes in our organoids after treatment, and this was seen after radiation and also in combination with radiotherapy with temozolomide, so the chemotherapy. And this was like the trend for most of the patients. So it was really consistent also across the patients that DNA damage response genes are activated after treatment. And then we did like additional validation on protein level. So we saw the results for gene expression before, but now we validate this for proteins. We choose like MDM2 and P21 as like the targeted proteins. We did the immunofluorescence before and after therapy, and also Western blot, and we noticed the same trend, so increase in those two proteins that are associated with P53 pathway, the cell cycle, the cell survival, and also apoptosis regulation. And this was all published by our postdoc in the lab, Bernarda Meitz. I hope I could give you like a glimpse of our research that was done specifically on this methodology. So targeted gene expression has been used to molecularly characterize patient tumors and also cellular models. We did like additional validation with correlation of publicly available databases or with the protein levels, and we could observe like similar trends. Some new biomarkers associated with poor prognosis of glioblastoma patients have been revealed, and we are working now further on these results to also functionally validate our biomarkers to see if they can be used as a targets, also not just a biomarkers, but also targets of therapy. And I show you some organoids work. So they can be really nice models that we show that they mimic the patient tumors, and they actually mimic the treatment response that we see in patients. And so we are now using these models to further evaluate some new therapeutic strategies, and especially the combination. So with chemotherapy, radiation, and then different targeted approaches, to see if they could be of use then for clinical validation. So at the end, I would also like to thank our support, so our European twinning cut cancer project that allows us to work deeper on those in this research. So if you're interested in our work, in some publication, dissemination that we are working on, you're welcome to follow the project. And I would like to thank all of the researchers and clinicians that are working on these projects, especially the cancer biology group and also phyto department for their technical help with the system and methodology. Our clinicians, so oncologists, radiologists, neurosurgeons, pathologists, also the computer science researchers that help us to correlate the data with the patient data and to make sense out of our data, and all the patients and donors and all the projects that supports our work and makes our research possible. So thank you for your attention. I hope I was clear, and please do not hesitate to contact me or members of our group if you're interested in collaboration. And I'm happy to take the questions.